Welcome to this week's CCI Conservation Seminar. Great to see a good turnout for this one. So everyone's really, I know I personally have been really excited for this one, and to welcome Bethan to give us a seminar here. We're welcoming Dr. Bethan Manley today from SPUN, the Society for the Protection of Underground Networks, which I think is one of the most catchy seminar titles we've had this term, and I expect for many of you the same as well. SPUN is an organisation that sets up for mapping and protecting the world's mycorrhizal fungi, a hidden link between biodiversity protection and carbon drawdown. That's what we're going to hear about from Bethan today. Bethan is homegrown, I might say. She did her PhD here at Cambridge on arbuscular mycorrhizal fungi and their genetic interactions with crop plants. She's went on to become a senior computer biologist at the Wellcome Sanger Institute, also here in Cambridge, and now works as a program manager for global data science at SPUN. And I've told Bethan she has a standing invitation to come and visit us here in CCI. I think there are a lot of potential collaborations that we could explore with many of the partners here and with the university. I also just wanted to highlight the context of, I think this is really timely talk as well, that Bethan's come to talk to us about mycorrhizal fungi, given that at the UN Biodiversity COP in Colombia, the proposal's been put forward for the fungal conservation pledge to incorporate funga alongside fauna and flora in multilateral environmental agreements, and thinking very much about funga, or the 3 Fs, when we're talking about conservation, recognizing that that's an entire kingdom. So with that, without further ado, I will hand us over to Bethan Manley. Welcome.
Yeah, thanks very much for the introduction and for having me here today. It's really lovely to be with so many relevant institutions and organisations. So yeah, I'm going to talk to you about SPUN's work, and I guess I don't really need to introduce myself anymore. My background. I'll tell you a little bit about SPUN just to start us off. So SPUN is a scientific non-profit, and we were founded by Professor Kobe Kiers, who is a professor of evolutionary biology in the Free University of Amsterdam. She founded SPUN about 3 years ago with this aim to map mycorrhizal networks and to advocate for their protection, because she was actually invited to do a TED talk on her research, and they asked her, do you have any organizations that you'd like to represent, or institutions other than your academic institution? And she just couldn't think of one that was advocating for the protection of mycorrhizal fungi. So that's kind of where the idea for SPUN came about. And I'll be walking you through everything that we work on today. Yeah, so as we said this morning, this is quite a timely discussion, because there was recently a proposal put forward to the UN Convention on Biodiversity that is advocating for fungi to be considered as a separate kingdom, because they are a separate kingdom from flora and fauna, yet they haven't really received a kingdom's worth of attention in conservation and science, really. So there's been quite a lot of really nice articles, a lot of media coverage about this proposal, if you want to check it out. But it sort of arose from this idea that there are actually 16 times more species of fungi than plants. At least we think so, because they haven't really been described particularly well, because they are quite difficult to describe. But there's 3 times less the funding and resources given to fungal research and to fungal conservation. And this is also just an image from Kew, and they bring out every couple of years, they bring out this really amazing state of the world's plants and fungi, which probably many of you know about. And they recently, this year, they did a sort of an estimation on the number of species of fungi that exist. And this is based on extrapolations from the number of species that we have, because obviously we have not been able to describe 2 million species. They're very difficult to describe, but I think there's been analyses that if we continue at the rate that we're describing fungal species, it'll take something like, I cannot remember the number, but it's like hundreds of thousands of years to actually fully describe all of the species of fungi that exist in the world. So at SPUN, we are not focusing on all of the fungi in the world. We actually have a particular focus, which is mycorrhizal fungi. And the networks aspect of SPUN really comes into play when you consider mycorrhizal fungi. They are network-forming organisms in the soil. And this is this image like this. This is from a New York Times coverage piece of SPUN in, I think, 2022, when we first launched. But I think it's a really good demonstration of mycorrhizal networks and how they form these really vast, extensive networks in the soil and link plants together and forage and have this really huge surface area for picking up nutrients. So they are symbionts. That's their whole deal. Symbionts of plant species. They actually live within the root systems of plants themselves, and they're really essential for nutrient and water uptake for plants, to an extent that plants actually can get pretty lazy and they rely almost sometimes 100% on mycorrhizal fungi, so they're mycorrhizal partners, for taking up nutrients and water. Mainly because these mycorrhizal fungi are actually better than them at taking up nutrients and water from the soil. You can just see it from the surface area. And they don't do that for free, however. So mycorrhizal fungi are obligate symbionts. They can't actually make their own carbon. They actually don't even have an enzyme for making lipids, which are pretty essential in life. So they get all of their carbon from their plant hosts. And that is the symbiosis. It's based on a nutrient trade. So not only are they absolutely prevalent in the soils, we know that they make up a huge proportion of living biomass in the soils where they're present, but they are quite difficult to see and to measure their contributions and to understand what they're doing because we can't see them. They're very difficult to track and to understand. However, there are some ways that we can visualize them and understand them, and this is a video taken from AMOF, which is the Biophysics Institute in Amsterdam. And this was produced from the people at AMOF and Toby Keys' lab at the Free University of Amsterdam. And this is not sped up. So this is actually in real time how the movement of nutrients and carbon looks in the soil all the time. I just think it's really, really incredible to just imagine that, there's kilometers of this under the soil all the time, just moving nutrients, moving, if you like, information, moving carbon, and we don't really fully understand the extent of what these networks are doing. So when we talk about mycorrhizal fungi, we're mainly talking about two main types. Sometimes when I give this talk to plant and fungal biologists, they get a little bit offended because there are a couple of other types of mycorrhizal fungi. So if you are one of those people, I'm sorry, I'm not going to talk about the other types. They're not as prevalent as these two. So I'm going to just focus on these two, which are the ectomycorrhizal fungi and the arbuscular mycorrhizal fungi. And the ectomycorrhizal fungi include really well-known fruiting body forming fungi like Amanita muscaria, which I'm showing here. So this is our really charismatic fly agaric mushroom, and it is ectomycorrhizal, which means that when you see one of these popping up from the ground, this is living in symbiosis with plants. It's not free living, it's not saprotrophic, it's not breaking down dead matter from the soil. It's actually taking all the carbon from a tree. So ectomycorrhizal fungi tend to be found in forest ecosystems. They're really, really prevalent in boreal and temperate forests because they tend to associate with tree species there. So I think that the statistic is that there's actually only something like 4% of all plant species form a symbiosis with ectomycorrhizal fungi. But those plant species are the ones that have lots and lots of biomass. So you get tons and tons of trees in a forest stand. So they are pretty prevalent just because they tend to associate with these extensive forests. And then moving on to arbuscular mycorrhizal fungi, which are the ones that I've spent most of my academic career working on. So I am a little bit biased towards these, but I think they're a little bit more exciting because you can never see them. So they're super mysterious. And what I'm showing in this picture here is what they look like when they're inside plant roots. So although ectomycorrhizal fungi do live inside the roots of plants. They form these sort of structures, like feeding structures around root cells in plants. But arbuscular mycorrhizal fungi are really cool because they actually make their feeding structures literally inside the root cells. So that's what you can see right here. That's a feeding structure that is taking over the whole plant root cell. And that's how important they are, because you can imagine, why would an organism let another organism literally fill up its entire cell? Like it must be extremely beneficial to have, that desire to just allow another creature like engulf your entire cell. It's really, really amazing. So this is what they tend to look like when they're living inside their host roots. And the reason I'm showing grasslands there is because arbuscular mycorrhizal fungi tend to be found quite prevalently in grasslands. They're very big fans of living with grasses, so the poaceae. But they also are found with many, many other plant species. So they actually can be hosted by 80% of all plant species in the whole world. So when you consider any plant species in the whole world, they almost certainly can host arbuscular mycorrhizal fungi. There's very few that can't. Yes, that's those guys. So unfortunately, as is the case with many organisms today, the biomes and habitats where mycorrhizal fungi are present are being lost, they're disappearing. We know that practices such as really intensive farming can be pretty harmful to them, which is quite intuitive because these practices tend to use fungicides. We know that, say, overuse of fertilizers is particularly bad for mycorrhizal fungi, because the plants then, they don't actually need to host mycorrhizal fungi because they're getting loads and loads of fertilizer dumped onto the field all the time. So it's all very readily available, and they actually shut down their interactions with mycorrhizal fungi, which of course would mean that in systems like the one I'm showing here, there just aren't mycorrhizal fungi present where there probably would have been before. And then of course, deforestation and habitat loss. We know that if you're destroying vegetation, if you're removing plant hosts, then of course by proxy you're going to be removing the obligate biotrophs that live with them, which are the mycorrhizal fungi. And not only are mycorrhizal fungi really crucial components of ecosystems, but they're also a really huge carbon sink. And we do know that billions of tons of carbon flow annually from plants to mycorrhizal fungi. This is obviously a key component of the interaction, that they're moving carbon into the fungal networks. What we still don't fully understand is how stable it is in the soil. And what we still haven't really produced enough data on is, what the extent of carbon added to the atmosphere is when you have things like deforestation, because we measure the loss of the trees, of the forests, but we're not measuring the loss when you're actually killing what could be 50% of the biomass of the soil, which is the mycorrhizal fungi. So this is all that mycorrhizal fungi are a really key component of ecosystems, just like this one, but they tend to be overlooked because they're very difficult to fully understand in complex systems. We can't see them, we can't measure them with observational data. I'm just going to let you look at this picture for half a second. It's very calming. Yes, a little reset. And then I will move on to SPUN's work, really. So these are our sort of three buckets of operation. This is what we tend to work on. And I'm going to go through each of them one by one, starting with this first one, which is mapping. So a lot of the time, of course, many of you will be very, very aware that to understand how organisms are doing and how they're reacting to changes in climate and temperature, all of this, you actually have to understand their distributions. And a lot of the time this is done with observational data, but we can't really do that with organisms that live in the soil. So this is quite a difficult task, but we have some ways we're approaching it, and I'm going to tell you about that. What we do is we build prediction maps. So of course, we can't go around, punching holes all through the soil in every single ecosystem, like every square meter everywhere, to measure which mycorrhizal fungi are present, which would probably be the ideal way of properly understanding, temporally and spatially where mycorrhizal fungi are present. what we can do is we can take data that we do have, and this is DNA sequencing data from soil samples, and we can extrapolate that to areas where we don't have data. And how we do this is by taking the world's largest database of fungal DNA, and this is from soil samples, and this is from Global Fungi, which is just a really, really incredible initiative. We wouldn't exist without them. They are an initiative based in Prague, and what they did is they brought together all of the DNA, well, environmental DNA sequencing data from all across the world, from soil samples, from leaf samples, from the ocean even, and they have given us access. They're actually freely available if you're interested in checking them out. And we take their mycorrhizal data only, and this is only soil data that we take. And we're able to get richness estimates from that sequencing data for each soil sample in the database. So we take these richness estimates and we pair it with these layers of geospatial data. And this will be a number of layers. So vegetation, climate, remote sensing data, precipitation, everything that we think might be relevant for understanding the distributions of mycorrhizal fungi. And then we have a random forest machine learning model that is tasked with exploring the relationships between these variables. So what layers, what variations in vegetation, in climate, in precipitation affect which species we're finding in the DNA sequencing data that we have. And then not only does it explore those, it uses that information to then extrapolate out all across the world so that for every square kilometre of the planet, we have an estimate on the richness value of mycorrhizal fungi for that square kilometre. And that's an extrapolation because we don't have a measure of mycorrhizal DNA richness for every single square kilometre of the planet. But what we do have, we're able to extrapolate out from to make a global map. And we actually produce two key maps, and that's because the two large groups of mycorrhizal fungi that we have are very distinct. They have very different reactions to precipitation, to climate, and to the hosts that they live with. So starting here, I'm showing the ectomycorrhizal prediction map. And this is sort of very unsurprising if you understand ectomycorrhizal fungi, because you can see them in these northern latitudes. In yellow, this is where we have particularly high richness of ectomycorrhizal fungi. And you can see that in the northern boreal and temperate forests, they have really high diversity up there, which really reflects where they tend to be found. There are a couple of patches towards the tropics, which is really cool to see because we do know that there are some forests in the tropics that are ectomycorrhizal hosts, where the trees are actually ectomycorrhizal, which is relatively unusual if you study ectomycorrhizal fungi. It's a very interesting area of study. But otherwise, it really does follow kind of what we'd expect for ectomycorrhizal distributions. And then this one here, it's almost kind of the reverse. These are arbuscular mycorrhizal fungi. And this follows a bit more of a sort of classic latitude distribution where we can see a lot of concentration of diversity towards the tropics. And again, that's not entirely surprising because we know that a lot of tropical plant species are the host of arbuscular mycorrhizal fungi. And I guess just intuitively, if you know that you have higher diversity of plants and 80% of plant species are able to host arbuscular mycorrhizal fungi, it makes a lot of sense that they tend to be found towards these in the tropics, where you might expect there to be higher diversity of plants. So it is important to mention here, though, obviously, that these prediction maps are predictions. So they're not absolutely based on empirical data for every single square kilometre of the planet. So we have to make extrapolations. We have to predict in many, many regions of the world. And what I'm showing here are the sort of is the extent of variation that we get between iterative runs of the model. And you can see that there are particular areas where we have quite high variation with the prediction, with the richness predictions that we get for every run. And it's the same for the extrapolation map that I'm showing here. So the model is kind of a bit strained in some regions because it's having to extrapolate wildly based on fewer numbers of samples. And that really is the problem, is that where you have fewer numbers of samples, you're more likely to have to work harder with the modeling to make wild guesses because it's just not based on as much data. And one of the causes of this, which I'm sure many people who work in ecology will understand really well, is sampling bias. So we tend to have more and more samples from areas where there's really high amounts of funding for science, and we tend to have fewer where there's reduced funding. It's a very key problem in a lot of ecology, and it just means that a lot of the time the data that we're working with global studies for any kind of organism tends to be based not really on like the actual biological reality, but on very biased samples. So one of the things we wanted to do at SPUN was to increase the number of samples that we have from those regions where we're having to extrapolate out wildly, or more than other regions. And we didn't really want to be the ones doing that ourselves, because we recognize that there are really serious funding imbalances in science. And a lot of the time, this does lead to researchers from the global north going to the global south to generate more data, and then not necessarily actually contributing to researchers and their work in those areas that they've collected in. So we launched in 2022 the Underground Explorers Programme, which is something we're very heavily invested in. And this is a grant funding program where we give small grants to researchers mainly from the global south, but because we're a mycorrhizal organization and we fund mycorrhizal scientists, there actually aren't that many. So the more that we have, the more we're able to fund people from outside the global south. But it's been a really, really wonderful program, and I definitely encourage you to find out more, because we just have some absolutely incredible scientists from all over the world who are mycorrhizal experts in ecosystems that are really vulnerable, that we don't have many samples in, and obviously they are the experts in these ecosystems, and they carry out their research programs that are just extremely well thought through and really sensitive to like the local environment and the people that live there. And it's just really delightful to work with them all and to support them to do this work. And that's one of the ways that we're generating more and more research on mycorrhizal fungi from across the world. And it's something we're still very invested in. So we have funding to do a couple more years of the Underground Explorers Programme. And we've already had 3 or 4 cohorts, I lose track, but it's been about 40 people per cohort. So we've just got tons and tons of people, and it's it's a great program. We love it. So moving on to the next aspect of our work. And so this is the sort of protect category, which is particularly relevant, I suppose, to the CCI and the work here. And this is sort of a push to incorporate fungi into global frameworks for conservation and protection of the networks themselves, to get them incorporated into biodiversity frameworks. So initially, I think a lot of people probably would question, or would be asking the question, surely if you're protecting above ground diversity, you also by proxy protect below ground diversity, which is a great question. And it was something that we obviously also wanted to address, because if we're able to have above ground indicators of mycorrhizal diversity, and then we can just focus on those, perhaps we don't need so many DNA sequencing samples, and we don't need to go and check places so thoroughly to identify mycorrhizal richness. So this is actually provisional work done by a SPUN member, Laura Van Galen, and she's just done this really incredible analysis on the above ground and below ground mismatches of richness between plants and mycorrhizal fungi. This is actually under review right now, so don't share it, but hopefully it'll be out at some point soon, and then you can all explore it. But what we were able to find from this work was that there is a positive relationship for both ecto and arbuscular mycorrhizal fungi with plant richness, which you would imagine, but it's actually not a particularly strong correlation. And what I'm showing in the maps is in green, you can see the 95th percentile, so hotspots of vegetation. So these are areas where we have really, really high diversity of plants. And it's the same in both the green. And then in purple, for the top, you can see in purple, this is the 95th percentile, so the hotspots of arbuscular mycorrhizal fungi. And in the bottom one, this is the 95th percentile of ectomycorrhizal fungi. And in pink, I'm not sure if you can see the pink super well. There are a couple of areas where we've zoomed in, but in pink are the overlaps between the vegetation and the mycorrhizal fungi. And there are plenty of overlaps, which does make sense, but there are also a lot of areas where we're just not seeing that correlation between there being enormous varieties of plant species and huge diversity of mycorrhizal fungi. That's particularly prevalent for the ectomycorrhizal fungi, which again makes sense because we know that they tend to associate with these northern latitude boreal forests where there's not as much species diversity. But it sort of highlights here that you may not always be able to take plant diversity as for mycorrhizal diversity. And of course, that previous work that I just showed, that doesn't actually show protected areas. So it shows diversity, it shows richness of plants as an indicator of mycorrhizal fungi and whether you can use that. But what I'm showing here is additional work that we've been doing to try and understand if protected areas are helping, if protected areas are overlapping with the areas that find hotspots of mycorrhizal fungi. So we've done an analysis where we took these prediction maps for both ectomycorrhizal fungi and arbuscular mycorrhizal fungi, used the world database on protected areas to see where we have mismatches or where we have overlaps of mycorrhizal hotspots with protected areas. And this is something we're exploring more and more. It's something we're very excited about because we want to see if the areas where we think there are crucial species or endemic species of mycorrhizal fungi are just like huge, hugely diverse, rich areas of mycorrhizal species, whether they're being protected and whether they're being considered under, or whether they're being protected. But what this doesn't do, and what we need to do more and more in future, is understand the actual impact of protection. Like if, are mycorrhizal communities flourishing in protected areas? We don't really know the answer to that because you can study above ground diversity in those areas. And we've done that, a lot of that. A lot of people have done work identifying how well populations of vertebrates, plants are doing in protected areas. But we have absolutely no idea if this actually does have a positive effect on the mycorrhizal communities, because they can be very affected by things like nutrient runoff from nearby farms, and the soil is quite a dynamic system. So this really has to be measured, and this is just sort of a step forward to understanding protected areas and the impact on mycorrhizal fungi. So I want to move on now to some of the work we've been doing on individual ecosystems and the impact and the role of mycorrhizal fungi on these ecosystems. So I'm including here some of the projects that we've done recently, and these projects are sort of with an aim of understanding unique contributions of certain mycorrhizal species or communities on maybe more vulnerable plant hosts or on environments that are under threat. So for example, we did a project in the Palmyra Atoll with the Nature Conservancy, and this was a really exciting project. They've been dealing with these invasive coconut trees on the Palmyra Atoll. If you don't know of it, I didn't really know of it before we started this project, but it's the most remote island chain in the world. It's very, very far away, and it's very vulnerable because of that. And it used to be, I think, a US Army base, so they had a lot of coconut trees planted there, and they've been really very negative. They've had a very negative impact on the native palm trees that were supposed to be there in the first place. And there's been a huge push to eradicate the invasive coconut trees. But we know that those native palm trees that were present in Palmyra Atoll were the host of these really incredible ectomycorrhizal species. So we've been monitoring whether this effort that they've had to reduce the number of invasive coconut trees has had a positive impact on the mycorrhizal communities that are present. That was a really exciting project recently. And then another example that I'm showing here is, can't really see it on there, but in the top, there's a project that we did in Italy in the Apennine Mountains. And this was monitoring mycorrhizal communities and how they were impacted by forest fires and devastating forest fires in the area. before I go into this. So this is a very relevant project. So this project, we carried out, this is I guess a more in-depth case study on one of our individual ecosystem level projects. And this was work that we carried out with the RSPB and ACBK, which is the sort of premier conservation organization from Kazakhstan. So we did this work with the Kazakh Steppe team, who are based here in this building. And we were, I think this was a particularly good case study for studying actually, and we had a lot of fun sort of conceptualizing it, because steppe systems are maybe not as charismatic or exciting visually as, you know, tropical forests, and adding a layer of mycorrhizal data to these systems is actually really important and really interesting, because arbuscular mycorrhizal fungi are just incredibly prevalent in grassland systems. And we know they probably play a very, very important role in, you know, buffering plants against water stress in these systems. And it's almost like these systems where it's less charismatic might benefit from, you know, an additional layer of importance and information from mycorrhizal fungi. So we had a lot of fun with this one. And a group from SPUN were lucky enough to go to the Kazakh steppe, and we sampled across many different steppe ecosystems, all the way from desertified steppe up to the Russian border, where there's feather steppe, I guess meadow steppe and forest steppe systems. And we took 56 samples, which was very impressive because this is a very large country. So it took a long time to travel that far. But actually previously in our models, we'd had no samples for the whole of Kazakhstan. So nobody had actually collected any soil samples and sequenced them specifically for mycorrhizal fungi in the whole of Kazakhstan. So we went from 0 to 56 in our models with data from Kazakhstan, which was really exciting for us. This actually led to a 1.3-fold increase in fungal samples from Central Asian grasslands, which is really beneficial for how the models predict. We found 92 mycorrhizal species that have been previously known to science, but we also found 200 species that we know are mycorrhizal because we're able to match them to databases, you know, at genera or maybe family level, but we don't actually have a species ID, which means that these organisms, they haven't been described. You know, we don't have a type specimen, we don't have a described species for that sequence that we identified. And I almost think that is almost more exciting than the previously described species. We often are able to match our data that we generate from specific projects to the IUCN Red List, because ectomycorrhizal fungi some of them are found on the IUCN lists. But in this case, as I mentioned, because grasslands tend to be really, really important for arbuscular mycorrhizal fungi, there are actually none of those on the IUCN Red List at all, which is partially because we can never see them above ground. So it's a difficult task getting them on there. But so we were not able to identify, in this case, any species from the IUCN Red List. And moving on, this is a little bit more of the mapping that we did for the project in Kazakhstan. And you can see that we predict pretty high AMF diversity across the whole of Kazakhstan. Again, this is because it has these huge swathes of grasslands. And we were able to really reduce our model uncertainty in certain areas because we were collecting more and more data. And that is really one of our aims, where we target particular areas to generate more data. And we were able to see that desertified and dry steppe systems had lower mycorrhizal diversity, which is very intuitive. You know, it makes a lot of sense that the drier, maybe less luscious areas don't have as many mycorrhizal fungi. But this is particularly interesting because the desertified steppe system is increasing over time. You know, as the steppe gets drier and more desertified, what we're hoping to see is also which species might be lost or increasingly pushed northwards, which we can do by looking at the differences between the species that are found in the meadow steppe, in the forest steppe, you know, all the way right down to the desertified steppe. And this is, I think, sort of relevant to what I was just talking about with the undescribed species that we identified in Kazakhstan. So we're able to make maps based on where we have species that have been identified, like where we have sequences that have been identified at species level, and where we have a lot of sequences that we just can't place at species level. And that's partly because of the state of databases for mycorrhizal fungi. There's a lot of difficulties there because you need type specimen to be able to register a species of mycorrhizal fungus, for everything. But that's very difficult when these are obligate symbionts that you can't necessarily culture very easily. Some of them are just possible to culture. They need their host plants there, and it's just very difficult if you don't fully understand what the individual hosts are for each one. You're able to describe them from the soil, but you can't isolate that and sequence it and morphologically identify it. So we're able to make maps where we have lots and lots of species that are unidentified, and we're able to see where we might have the most impact on taxonomic studies. So, supporting culture collections, this is something we're really hoping to get into, is species discovery and, maybe supporting culture collections. But it's also helpful in allowing us to see where we might have the most impact in sampling priorities. So looking at areas where we have not as many species that have been identified. And yeah, this is particularly interesting because 88% of our sequences in our whole data set are not assigned at species level. It's still useful, we can still see that they're mycorrhizal fungi because we have maybe genera level assignments, but there's just so much work to do with species discovery. And this is another one of our sort of targeted approaches where we're hoping to see whether there are indicators above ground of mycorrhizal richness. And again, this is a fairly intuitive analysis because we looked at forest age and how that impacts mycorrhizal diversity. And you can see that, of course, these old growth forests, these really ancient forests, have really, really high mycorrhizal diversity. That was kind of interesting, though, because we were seeing that there's quite a big jump from even the age, the age bracket of 100 to 150. So it does seem like there's just a huge jump from even what you would consider older forests to these ancient and old growth forests. And that's particularly interesting because, well, actually what I'm showing here is just an overlapping of forest age with mycorrhizal diversity, which is how we were able to get this analysis. And this is particularly interesting because a lot of the time when we consider ancient forests, we're thinking about the carbon that they store, their huge carbon sinks, and it has to be considered, the fact that these are also hugely diverse mycorrhizal communities, and they're probably playing a huge role in taking that carbon from these ancient forests and putting it in the basin. And we don't fully understand how long they keep it there or what they do with it when it's in the soil. It's one of my final protect aspects, and this is something that we're doing to understand the threats to mycorrhizal fungi. So when we think about threat mapping, this is sort of mapping the intensity, the distributions of threats to organisms. And when you consider mycorrhizal fungi, there are particular threats that are probably more obvious or more prevalent or more likely to impact mycorrhizal fungi than, say, other organisms. So some of the threats that we've identified are intensity of agriculture, climate changes, habitat loss, soil pollution is obviously a really big one. We're able to combine all of these geospatial layers and create threat indexes for mycorrhizal fungi. And the threat index that's been created shows actually that it does actually correlate very, very well with mycorrhizal diversity in comparison to, for example, to nematode abundance or plant abundance, because obviously a lot of these threats do affect many organisms. But the threat index that we've been able to build is particularly well correlated with mycorrhizal diversity. And information like this allows us to sort of overlay with our richness maps, which is this map of Colombia that I'm showing here, to see where we might have the most threatened populations of mycorrhizal fungi. So where do we have high concentrations of diversity of fungi, mycorrhizal fungi, and high threats? So where are networks most vulnerable? So moving on just to our final bucket of work that we do, and I know this has been kind of like a whistle-stop tour through a lot of SPUN's work, so I'm going to be brief with the last one because it's also the hardest. But we want to not just protect mycorrhizal fungi themselves and, for the sake of it, although we do believe in protecting biodiversity for the sake of it. We also want to protect their ecosystem contributions because they really do have a lot of ecosystem contributions. They're just absolutely crucial. So some of the work that we're doing is in trying to A, understand their functions in specific ecosystems, and B, protect them, maintain it. This is a project that I just wanted to briefly mention, which is work that we're doing with the JGI, or the Joint Genome Institute, in collaboration with INRAE in France and Universidad del Rosario in Colombia. And this is an effort to understand the function of ectomycorrhizal fungi, in this case, in specific forest systems in Colombia and Corsica. And Colombia and Corsica are both ectomycorrhizal. The forests that we're studying there, they're ectomycorrhizal dominated areas. And they're particularly interesting to us because the sites that we've identified are under particular threat from deforestation or climate change. And what we're doing is trying to understand through generating genomes and through metatranscriptomics work, the actual specific mechanisms that are contributing to these systems. So we can do this by generating genomes so that we're populating databases, sequencing the genomes of mycorrhizal fungi actually lets us, it's more likely to get us closer to species discovery because we can register them. We have type specimen in these cases, and I'm showing our expeditions lead here, Adriana Corrales, who's a tropical ectomycorrhizal expert. She's great at IDing them and getting morphological type specimen. We generate genomes for these, which populates databases and allows us to have species discovery. But what we're also doing is we're carting out huge bucketfuls of dry ice into the field, which is particularly difficult for Adriana in Colombia, maybe a little bit less so in Corsica, to basically freeze soil samples, snap freeze them, and to sequence the RNA from these soil samples. And this is so that we can actually see which genes are active in the ecosystems that belong to mycorrhizal fungi. So, how are they contributing to the nutrient turnover in these systems? Because that's something they're really, really important in is nutrient cycling in these ecosystems. Or maybe how are their enzymes operating that break down carbon compounds, or contribute to the loss into the soil. And then this is a kind of a final aspect of our ecosystem function work, which is understanding the impact on plant productivity. Because these are obligate symbionts, they have obviously one of their biggest contributions to ecosystems in how they interact with host plants. And what we're able to do is look at the impact of mycorrhizal fungi on an ecosystem by looking at how they impact their plant partners. So we've been able to sort of correlate fungal diversity with net primary productivity, which is a geospatial layer that we're able to have access to. And this, again, at the moment, this is a correlation, but we're able to sort of explore how they impact the net primary productivity of plants. And the more research we have available for this, the more we'll be able to understand. But we're also able to do sort of future forecasting to look at how much productivity we might lose in plants with the loss of mycorrhizal networks and with the loss of their mycorrhizal partners that might be, for example, buffering them against water stress or helping them access nutrients, particularly in nutrient poor conditions, which we know are particularly important for mycorrhizal interactions. And then finally, obviously our sort of one of the most poignant aspects of mycorrhizal function is just their sheer capacity to bring down carbon into the soil from their plant hosts, and how at the moment this is really still kind of a black box in research. So SPUN was a contributor on a paper last year which identified that 13 billion tons of CO2 is traded from plants to mycorrhizal fungi every year, which I think the stat is that that amounts to about a third of anthropogenic emissions every year. So we know that they're moving this carbon into mycorrhizal networks. That's been kind of extensively measured, but it's still, it's still sort of a statistic that needs to be tested and tested. It was analyzed across lots of different plant hosts, across lots of different systems. So it's still a provisional statistic, but it's a start. It's difficult to measure. So we do know that a lot of carbon is moving into mycorrhizal networks. It's kind of a fundamental aspect of the symbiosis. but it's just so it's such a complex environment down there. How stable the carbon is, how like the interactions between mycorrhizal fungi and other soil microbes and how that causes it to get broken down. Necromass, dead mycorrhizal networks, how they keep it in the soil. This is all such a black box to us. And it really does need to be investigated, because this is huge amounts of carbon that just aren't really being accounted for. And they need to be properly understood and measured before we're losing these networks. So with that, just back to this image. Just hopefully, not back to that one. So hopefully I've kind of convinced you that above ground diversity is really reliant on below ground diversity, particularly mycorrhizal fungi. And although things are getting better, like we've sort of discussed in fungal conservation, we would still probably argue at SPUN that there is an above ground bias towards conservation, even in the fungal kingdoms. A lot of the time, the fungi that we measure and understand in conservation contexts are the ones that form these above ground fruiting bodies. So with that, I will go on to my thank yous, which are, of course, to the SPUN team who produce all of this data and go on the expeditions and do all of this amazing analysis. The global fungi team who give us the data and work with us so closely on gathering together and curating their amazing resource of eDNA data. The Crowther Lab at ETH Zurich, who produce the random forest models that are able to do our prediction maps. RSPB and ACBK, who are just excellent partners on the Kazakhstan expedition, and many others. Yeah, and I'm happy to take questions. Thank you.
Thanks so much, Bethan, for a really fascinating seminar. I think everyone will agree. I think we've got a few minutes for questions, and I'm sure there will be a few questions. If I can steal 60 seconds for just some reflections. I was really struck by some of your maps of protected areas in relation to mycorrhizal fungal diversity, and I think a figure you had in there that 90% of mycorrhizal fungal biodiversity is not protected. I think one of the maps you had in there was it's like the fungal biodiversity hotspot was almost perfectly avoided by the protected area network. It's really incredible. So that's really important to take into account. Also taken on the message on behalf of IUCN, I know James can be here today, but we have at least one person really banging the drum in the IUCN red list to get those fungal red list assessments and really ramp that up. But I'm not sure, it sounds like we really need to focus also on the arbuscular mycorrhizal fungi as well as the ectomycorrhizal fungi as well, recognizing that there are just 4 key biodiversity areas, which we were talking about earlier. Several of us work on those. There are just 4 globally recognized for fungi currently out of 16,500. So that's another area where I think we could really synergize with you to identify those important areas that need protection and conservation. So really looking forward to working with you in those areas. I'll open it up for any questions. I don't know if there are any online. Diane, not yet.
Not yet.
Yeah, Eddie.
Hi, thank you. Really great talk. I work for Forestry England, which looks after the forest estate in England, and we've had hundreds of eDNA fungi samples, which we've made open access, but I want to make sure that you are aware of and you're using in your maps. My question, though, is about tree planting. There's a lot going on, particularly in this country. And one of the reasons that I hypothesized for why that often fails is that they don't have the fungi available in the soil because it may be degraded agricultural land, for example, and it just doesn't have the right fungi for trees. There's quite a lot of companies popping up which are selling, supplements, if you like, that you can plant alongside your tree, and they're the key mycorrhizae. What's your reflections on that?
Yeah, it's something that we're increasingly having to deal with. So it is something that has to be dealt with. It's just a difficult question. So like you said, sometimes we have been doing projects with restoration initiatives, and that is a big question. And it does make a lot of sense that if you are actively reforesting, you should probably actively repopulate the soil microbiome, especially, in temperate regions where we know ectomycorrhizal fungi are particularly important. The trouble with a lot of companies, mycorrhizal inoculum companies, so they do tend to, there isn't any regulation, so they do tend to just put lots and lots of different spores from generalists into, you know, an inoculum and spray it on. And as a species, we have done a lot of moving organisms around and dealt with the consequences of that. And so mycorrhizal fungi can be invasive. So, for example, when people planted lots and lots of pines in Australasia, they also were finding that they couldn't plant pine trees without mycorrhizal fungi, so they were bringing soil down. There's actually quite a lot of species of mycorrhizal fungi in Australia that shouldn't be there, and that are, some of them are poisonous, and people don't always know that. And so mycorrhizal fungi can be invasive. They can cause problems to the native mycorrhizal fungi, and we would definitely advocate for returning mycorrhizal fungi to ecosystems where they should be there. But I think that's why we're pushing so much to understand mycorrhizal distributions, because if you know where something should be or where it is usually, then you can probably more actively understand what you should be putting back into a system that's degraded, rather than just putting generalists that tend to grow quickly or, you know, establish really, really fast. It is one of the big questions that we're facing at the moment. It's very interesting, but we want to generate more data on which species should be where, basically, and use that to inform how to properly restore mycorrhizal networks to areas where they should be. And also with the eDNA, Global Fungi has everything that's publicly available. So if you have published data, if it's published or available, we will have that, which is good.
Questions, one at the back, and then Sylvia.
How are you, I'm in Tanner, plant sciences. How are you defining the species of VA mycorrhizae? And is it comparable with, say, a definition of above ground biodiversity?
Yeah, it's a great question. So it's kind of different between arbuscular mycorrhizal fungi and ectomycorrhizal fungi. So with ectomycorrhizal fungi, we use the UNITE database. I don't know if you know it. It uses species hypotheses, which are, of course, like sort of clusters of sequences at 97%. Most of them are based, a lot of them are based on type specimen and sequences linked to type specimen. That's all we can do. So that's how we define a species, like a described species. Arbuscular mycorrhizal fungi are even more difficult. They cause us so many problems all the time. They are present in UNITE, but we actually have to use a different metabarcoding region to identify them. UNITE only hosts ITS if you're a metabarcoding person. And so we have actually curated databases for arbuscular mycorrhizal fungi, again, which are based on type specimen, but they are partially based on eDNA data. So this is all to say, it's partially based on type specimen, linking to type specimen, also partially based on existing eDNA data that we know clusters at 97% with those type specimen and where they're found across the world. But yeah, it's challenging. So yeah, it doesn't always match up to how above ground diversity is classified. But yeah, it's just one of the challenges of the field, unfortunately.
Thanks, Ed. Great question. Sylvia, and then we might have time for one more if you keep it brief.
I'll try and be very quick, but fascinating talk. Just very quickly, actually, you were pointing out that there's a divergence between the plant diversity and the fungal diversity. And I was wondering, is there a sort of critical factor that influences that divergence? And if there is, are they different between temperate and tropical regions?
So I guess yeah, it's a good question. So with ectomycorrhizal fungi, the divergence is kind of obvious because they tend to be found in the northern latitudes, and the highest concentrations of plant richness tends to be not in the northern latitudes. So that's part of one of the reasons. That one's quite obvious. It's quite difficult to say with arbuscular mycorrhizal fungi because they tend to be able to associate with multiple plant species at the same time. We do know also that they're impacted by temperature and precipitation differently to plants. So I guess it's not necessarily entirely surprising that there are some mismatches. The other thing, of course, is just that, you know, these are all exploratory analyses, and we're taking 95th percentile diversity richness of plants produced from a different group to us, you know, and then we're comparing it to our 95th percentiles. These are kind of the ways that we're able at the moment to explore this data and to see, you know, what the state of mycorrhizal hotspots and where they overlap with above ground diversity are. But yeah, it's still an exploration. So yeah, and that part of the work is not my expertise, unfortunately, but I can always link everybody to the paper on when that comes out, because it's almost ready. So yeah, it's been a nice analysis. It's very exciting.
Thanks, Bethan. I think we can squeeze in one more question.
Are the presence of fungal biodiversity or biomass, in your opinion, visible for remote sensing technologies?
That's a great question, and we wish it was. I mean, we'd love that because it would make our lives so much easier. There is literally one potential way that we can do it, and that is plants produce different compounds in their leaves when they are associated with arbuscular mycorrhizal fungi and with ectomycorrhizal fungi. So there is work being done to see if we can identify, because we don't actually even know all the time which plant hosts are associated with which, because some of them can do both. And we don't know for every single plant species in the world if they're hosts of AMF or ECM. So that being able to identify based on leaf compounds, which you can do with remote sensing, there's a sort of not the best at remote sensing, but you can do sort of flyby remote sensing and identify these compounds. So you can technically see from the air which mycorrhizal group is the most prevalent in the vegetation that you're surveying, which is useful information, but very early stages, and it's not, you can't do it from satellite data because that's not powerful enough to identify these compounds in the leaf. That's really the only way that you'd be able to do it from remote sensing, unless you had an above ground proxy, you know, some kind of indicator for mycorrhizal richness, which again is very difficult to come by.
Thanks so much, Bethan. Yeah, thanks so much for coming. If you do have any questions that you weren't able to ask because we have run out of time, I know Bethan's going to stick around for a little while afterwards. So I think that all that's left for me to tell you is that next week's seminar will be same time, same place with Professor David Edwards. So they've joined, came to Cambridge last year as Professor of Plant Ecology, and I think this is his first CCI conservation seminar. So it'll be a great chance for some of us who haven't had a chance to meet Ed and hear about meet Dave and hear about his work to hear about that. So same time next week. Thanks everyone for coming. We did record this seminar, so if you want to go back and look at some of those lovely maps and recatch any of that, then you should be able to do so on the CCI YouTube channel in probably a few days' time. So just once more, thanks again so much, Bethan, for coming along and talking.